All life is classified into three domains: archaea, bacteria, and eukarya. We have prokaryotes and eukaryotes. Prokaryotes are organisms with no nucleus, and they're going to be the domains archaea and bacteria. Eukaryotes are organisms with a nucleus, and they're the domain eukarya. If you remember back from biology, the levels of classification start with domain, and then it goes kingdom, phylum, class, order, family, genus, and species. You might remember from biology making a mnemonic for the levels of classification, like King Philip came over 4 great spaghetti, so you could remember them in order. So our prokaryotes are found in 2 domains, archaea and bacteria. We're going to start with domain bacteria. In domain bacteria, you have kingdom eubacteria. This is common bacteria, including the 5 phyla, which includes Proteobacteria, Cyanobacteria, gram-positive eubacteria, spirochetes, and Chlamydae. Their cell walls contain peptidoglycan, and they're found pretty much anywhere. Domain archaea has the kingdom Archaeobacteria, which are ancient bacteria, including 4 major phyla: Euryarchaeota, Crenarchaeota, Nanoarchaeota, and Corachaeota. Their cell walls contain only 10% peptidoglycan, and they're mostly extremophiles. Since prokaryotes do not have nuclei, their genetic material, which is either DNA or RNA, is found within the nucleoid region. We need to know the following prokaryotic cell structures. We're going to start with the cell envelope. In the cell envelope, you have the cytoplasmic membrane, which surrounds the cell. It's the barrier between the internal and external environment. Then after that, you have the cell wall, which is a rigid barrier that encloses the cell membrane. It acts like a tight corset that keeps all of the cell contents inside and gives it a more rigid structure. Then you have the capsule. This enables some bacteria to adhere to certain surfaces, and it allows them to hide from the host immune systems and being able for them to cause disease. Eubacteria have peptidoglycan in their cell walls. Gram-positive bacteria have a larger ring of peptidoglycan than gram-negative. This makes the bacterial cell rigid and strong. Gram-negative bacteria have an outer membrane, a unique lipid bilayer that's made up of proteins. It serves as a protective barrier, excluding many compounds that would hurt the cell, including antimicrobial drugs. This outer membrane's outside edge is made up of lipopolysaccharides and endotoxin, so it's called the LPS layer. So here you can see the difference between gram-negative and gram-positive cell walls. So in gram-negative, they have that extra outer membrane that's on the outside layer of their peptidoglycan layer, which is very thin. Compared to gram-positive, their peptidoglycan layer is very wide. However, they don't have that extra external outer membrane. Hopefully you remember when we talked about the gram stain last unit. When you do the gram stain to a gram-negative cell, it's going to turn that reddish color. And when you do the gram stain, a gram-positive cell is going to turn that purplish color. Penicillin is an antibiotic that interferes with peptidoglycan synthesis, so the bacteria can't produce that peptidoglycan to make that layer. It's more effective on gram-positive cells because the outer membrane of gram-negative cells are harder to penetrate. Lysozymes are enzymes found in tears and saliva, and they break the bonds in the backbone of the peptidoglycan molecule, so that can be used to break down the cell walls as well. The capsule of a bacterial cell is a distinct gel-like layer used for protection or to attach to surfaces. It makes it resistant to washing off. The slime layer is an irregular gel-like layer used for protection or attachment, and it easily washes off. They can grow as a biofilm, a polysaccharide-encased mass of bacteria coating a surface. For example, dental plaque is caused by Streptococcus mutans, and it's called a biofilm because it's biological organisms, right? It's all of the bacterial cells that are growing together as one film on a surface, which would be your teeth. By having these defense systems, the bacterial cells can cause disease, like Streptococcus pneumoniae, which gives you pneumonia. Over here on the right, you can see the parts of the bacterial cell, the main parts. You have the nuclear material found in the nucleoid region, because remember, they don't have a nucleus. You have your cytoplasmic membrane. Then you're going to have a cell wall. This is a flagella. The cytoplasm is like the gel-like material in the middle. And the slime layer or a capsule would be on the outside here. So a bacterial cell could have a number of different external parts. So parts that extend beyond the internal part of the cell. Examples would be the flagellum, fimbriae, or pili. A flagellum is a long protein structure used in movement. It's made up of 3 parts: the filament, the flagellum, and the basal body. Some bacteria have one flagellum, while others have many flagella. Fimbriae and pili are interchangeable terms used to designate short, hair-like structures on the surfaces of prokaryotic cells. Fimbriae have nothing to do with movement. There are exceptions, but fimbriae are most often involved in adherence of bacteria to surfaces, so it allows the bacteria to hold on to something. In E. coli, a specialized type of pilus is called the F or the sex pilus, is used during mating in a process called conjugation. And this is actually sexual reproduction. They are actually exchanging genetic material. Common pili, almost always called fimbriae, are usually involved in specific adherence or attachment of prokaryotes to surfaces in nature. In medical situations, they are major determinants of bacterial virulence because they allow pathogens to attach to or colonize tissues and or to resist attack by phagocytic white blood cells. For example, pathogenic Neisseria gonorrhea adheres specifically to the human cervical or urethral epithelium, which are the skin cells, by means of its fimbriae. So here you see the flagellum and all of its parts. So you've got the basal body here. That's what anchors the flagellum to the cell wall. You got the filament that comes out, and that's what's used for movement. And then the flagellum is the protein subunits that make up the filament. So they're found inside the filament. Then you have pili and fimbriae over here. This one is what that pilus was called, the sex pilus or the F pilus, where they actually use it for conjugation. So it's sexual reproduction. And then this is fimbriae on the outside of a bacterial cell that's being used to attach that cell to a surface. Let's look at some internal bacterial structures. So we'll start with chromosomes. These are irregular masses found within the cytoplasm that form a gel-like region called the nucleoid. Usually a single circular double-stranded DNA molecule contains all of its genetic information. It's usually twisted very tightly, called supercoiled. Plasmids are circular supercoiled double-stranded DNA molecules. They're smaller than a chromosome. One cell can have many different plasmids. The cell does not necessarily need the genetic info carried on the plasmid, but it can use it when it needs it. The ribosomes are where protein synthesis takes place, so that's where the proteins are made. Prokaryotic ribosomes are 70S in size, composed of a 30S and a 50S subunit. The higher the S, the greater the density and the faster they move. In comparison, eukaryotic ribosomes are 80S. Differences in ribosomal structure serve as targets for certain antibiotics, which bind to the 70S ribosome and inhibit protein synthesis in the bacterial cell. Endospores are dormant cells produced during sporulation in certain bacterial genera, like Bacillus and Clostridium. They can remain dormant for more than 100 years. They're resistant to heat, toxic chemicals, and UV radiation. The surviving endospores can germinate or exit that dormant stage to become a typical vegetative, which means actively multiplying cell. Botulism results from ingesting endospores of Clostridium botulinum that become vegetative in the body. So they were dormant before you ingested them, and then in your intestines, they became vegetative. They exited out of the dormant stage, they germinated into a vegetative cell. Symptoms include vomiting, dry mouth, weakness, abdominal cramps, difficulty swallowing, speaking, and breathing. So how does an endospore actually germinate and turn back into a vegetative cell? Well, the endospore is triggered by either heat or certain chemicals. The endospore takes in water and swells up, causing the spore coat and the cortex to crack open. The vegetative cell then grows out. There are many different shapes and arrangements of bacterial cells or clusters of cells. So coccus are going to be round, rods are going to be bacillus, curved forms are going to either be spirillums or spirochetes. Diplococci means you have cocci in pairs, so two of the round ones stuck together. Neisseria is coffee bean shaped in pairs, so they're coffee bean shaped and stuck together. Coccobacilli is going to be your bacillus, so the rod shaped ones that are together with the coccus one. Vibrios are curved rods, so they look like rods but they're curved. Tetrads are going to be coccus, which are the round ones that are in packets of 4. Sarcinae are cocci that are in packets of 8, 16, or 32. Mycobacteria look kind of like stringy spaghetti. Corneobacteria are in this, what they call a palisades arrangement. Spirilia are going to be like spiral looking. Streptococci, so strepto means in a chain, and cocci means the round. So streptococci are a chain of round cells. So streptobacillus would be a chain of the rod-shaped cells. Micrococci and staphylococci are large cocci that are in irregular clusters. A cluster means that it's like stuck together, but in this more of an irregular shape. So staphylococcus is staphylo, meaning the cluster, and coccus, meaning the round circles. Spore-forming rods look like a rod that has a spore on the end. Streptomycetes look like moldy and like filamentous looking. And spirochetes are also look very spiral. So take a couple of minutes and practice labeling these different bacterial shapes. Okay, let's see how you did. So these are just individual circles of bacterial cells, and they are called single cocci. These are going to be round cells, but they're in a chain. So in a chain, they're called streptococci. These are round cells, but they're in clusters, so like in different irregular shaped clusters. And so they're called staphylococci. This one over here is a single rod shaped, so it's going to be called a single bacillus. This one is 2 rod shapes, so it's called a diplobacilla. This one is a chain of rod shapes, and so it's called streptobacilli. And this one is going to be 2 that are together, where you have a coccus, so a round one with a rod one. So you can see like there's going to be one end that looks like a round ball and then one end that looks more like the rod, and that's Coccobacillus. And lastly for today, we're going to go over cyanobacteria. They're a species found in the eubacteria kingdom. They're ancient photosynthetic bacteria, also called blue-green algae. They're one of the Earth's top producers of oxygen. They are the ancestors to what we know today as algae and plants. They're found in almost every terrestrial and aquatic habitat, so oceans, freshwaters, bare rock, soils, and even in the fur of sloths. They perform nitrogen fixation, So they fix the otherwise unusable atmospheric nitrogen into a usable form for the plants. Some pros of cyanobacteria are the fact that they produce so much oxygen for the environment. They fix nitrogen for the plants. They're important for marine and terrestrial food chains. Possible antiviral applications can be found by studying them. Possible dietary supplements can be found by using them. Some cons include blooms of cyanobacteria due to nitrogen-based runoff, either from agricultural fertilizer or from waste, have been responsible for killing aquatic life, making the waters hypoxic, which means oxygen depleted, in the Gulf of Mexico, off the coast of North Carolina, and elsewhere. They're potentially harmful to people and animals in the right concentrations. So these are a couple different genera of cyanobacteria. So in A, you have like Gloeocapsa. They're called glow caps because the yellowish hue given off by the caps. And then you have B, Oscillatoria, which are filamentous bacteria known for its oscillating motion.